I'm coming to you from Brisbane, Australia, talking to you about our uncoupling pathways involved in immunotherapy resistance. I'll talk about some of the work that the lab is doing at the cross section of cancer and infectious disease. I lead the clinical omics lab at the Fraser Institute at the University of Queensland, and we've recently set up the Queensland Spatial Biology Centre. And so I'd like to start by acknowledging the funding agencies. Without them, none of this work would be possible, and also Bruker for the kind invitation to present at this meeting. Let's go forward. Perfect. So spatial biology is a really exciting field, and it's giving new insights into disease biology. We've written a couple of pieces for GenEng on how it's involved in the top 10 spatial biology companies, how it's redeveloping and redefining the multiomics approach, how it's reorientating biopharma development. And we've got a number of studies, which I'll go into a few of them, looking at the impact of COVID on the lungs, the hearts, the brain, the placenta. Really exciting studies looking at where the virus is localizing to in multiple organs and the effect and contrast between COVID and flu and multiple pandemics. And we've also written a more recent piece on sort of the field and where it's going towards. I'll highlight a number of studies where my lab has contributed to efforts using the NanoString geomics platform, which is, these are some of the original research papers, using the geomics platform in head and neck cancer. We've got a couple of studies in lung cancer, a couple in breast cancer, and more recently in sort of classical Hodgkin's lymphoma. And so these are ongoing active studies, of which we've published a few of those. And this is now looking at the studies we've published in COVID-19, looking at the impact of COVID on the lungs, the heart, the placenta, really large team studies, and sort of expansions into computational approaches of multi-omic data integration. Standard, which is a package which was developed by Chinwei and Ning and Mel Davies' team on data normalization, QC, and then also the DE analysis and sort of a biomarker triage biomarker study, which was a multi-cohort study led by my lab. And also more recently, really shifting gears into the NanoString Cosmics platform, which is giving us single cell resolution and workflows that we've established for both analysis of spatial neighborhoods, but also applications to localize signal down to single cells. And we have some manuscripts in prep at the moment. And so I'm really excited by this renaissance in Bruker spatial biology. It's really showcasing how these platforms are coming together, multicellular high throughput and high plex single cell resolution, and also the complementarity of whole slide proteomics and DNA analysis. So you're getting the full gambit of RNA, protein, DNA, all within the Bruker infrastructure and within the multi sort of platform setting. And so I like to say that spatial takes a village. These are sort of the approaches my lab uses in both the transcriptomics and the proteomics space. Traditional biomedical dogma will tell you that you should do transcriptomics upstream, whole transcriptome, and then go validate using spatial proteomics. But what's happened in the last sort of couple of years is that as we've really improved on our plex levels with the protein, we're now looking at 50, 60, 100 markers. The spatial proteomics also becomes potential discovery too. And so how this comes together is this computational biology or spatial informatics approach, which is, 10 years ago, you would send data to one bioinformatician. Today, these are complex data sets of molecular readout simultaneously with pathology, annotations of tissues, and bridging this is this sort of spatial informatics space where we really need a really collaborative effort to understand the tissue architecture at the molecular level. So we collaborate with multiple partners in this space. And then it's really exciting to sort of be across the breadth of these assays, whether it's 3D biology, whether it's proximity ligation work, interaction work, and so on. And so the lab focuses on immunotherapy, predictive biomarkers. This work's really led in the field by what's been done in melanoma, non-small cell lung cancer, head and neck, and so on. And this paper came out in Nature last a couple of years ago, and it showed that tissues and not blood are where immune cells function. And it was a really important paper because it showcased why we need to look at the tissue simultaneously with molecular readout to really glean insights into the microenvironment. Remember, your single cell experiments are looking at 10 to 50,000 cells in total in a single tissue section from a cancer biopsy or surgical resection. We're looking at 3 to 5 million cells per sample. So every time we run a spatial experiment, it's 20 to 50 times that single cell experiment. So the complexity of the microenvironment is a lot more, and what we need are true spatial single cell solutions to really understand them. So there are a number of predictive biomarkers for this: tumor mutation burden, PD-L1 expression on solid tissue. And what my lab tried to do is to do a deep dive on the microenvironment to profile every cell within those tissues. So 3 to 5 million cells per sample and map every cell within that. And why this is important is if you look at these two tissues at the bottom left, you'll see the one on the right is highly infiltrated compared to the one on the left. But if we were to grind these two tissues up, they'd have very similar TIS scores or tumor infiltrating scores. So getting that visual data is really important. And at a bulk level, we miss all that infiltration information. And so spatial is allowing us to get deeper insights into, this is just on a Kaplan-Meier curve, but we can see how by just using the distance metric of 30 micron from the CD8 T cells, we can glean a lot more information into the subsets that are actually predictive of clinical benefit. So in lung cancer, this is in second line IO treated cohort. We looked at a number of patients that were given immunotherapy. In the second line setting, we had multiplex IF data as well. And we asked the question, using an unbiased lens of discovery, could we gate on tumor and stromal features to identify predictive signatures associated with clinical endpoints such as progression-free survival and overall survival? And we used the Cancer Transcriptome Atlas panel. We've also used the whole transcriptome and a targeted proteomic panel. And we gated on cytokeratin to liberate the tumor mask and non-cytokeratin to liberate the non-tumor mask. So this was prepared in a tissue microarray, single core biopsy per patient. And we sample the tumor and the microenvironment. And so we could then identify at the RNA level in the tumor compartment between the responders and the non-responders the differentially expressed proteins and mRNA between the two groups. And we could also look at the stromal compartments. And what we found in this study was IL-2 and SPP1 osteopontin were highly enriched for in the responder group. And so when we looked at the contribution of IL-2 almost by itself within the stromal compartment identified the patients that would benefit from checkpoint inhibitor therapy in the second line setting in lung cancer. And it provided us a lens in which we could potentially validate some of these findings in orthogonal cohorts. We then layered this with multiplex IF data so we could look at compositional differences in the individual tissue arrays and start to contrast the co-expression of markers as you move into the tumor and as you move into the microenvironment. And so what we found was that we could identify 9 different neighborhoods in this cohort, not at the single core level, but at a cohort level. This is single cell proteomic data now. We could identify 9 different neighborhoods at 6-plex multiplex IF data. But the challenge with these data sets is we're binning a lot of these cell types that as other. And so really to get information from this, we could identify these individual neighborhoods, and then we could contrast between the non-responders and the responders. And we found this CD68 PD1 FOXP3 population that was highly enriched for in the non-response group. And so this was at 6-plex, and we wanted to get a lot more information. And so we built a panel of about 50 markers spanning tissue architecture, immune infiltration, immune profiling, lymphocyte, and this custom module, which involves enzymes against metabolic activity that is allowing us functional insights into cell phenotype. So we can overlay functional data over cell phenotype alone. And this is the computational pipeline for this. And what we found, and this is sort of James led this study from the lab, we can do the multiplex staining, we can do the image processing, the tissue QC for the areas of artifacts, fragmentation, necrosis, and so on. We do staining QCs, and then we cell phenotype. So we cluster based on about 50 markers per cell. We can identify multiple different layers of immune cell decons, and then do all the spatial feature analysis and comparative sort of clinical analysis on this. And so we can start to annotate over these individual cores the composition. Now you're going from a 6-plex to a 50-plex. We can get more granular about the responders and the non-responders. So we can see where the tumor cells are, the proliferating tumor cells, the stem-like tumor cells, the HLA-DR positive tumor cells, the B cells, the CD4s, the CD8s, and so on, per core across the entire cohort. And then we can ask the question, what are the differences between the responders and the non-responders in compositional differences between the two groups? And we find that there's this increased Treg population in the non-responders. And then we do what's called nearest neighbor analysis, and this is identifying the individual neighborhoods within the microenvironment. And we find this macrophage mixed tumor phenotype in the response group. And so we can use these layers of imaging data to now understand insights into cell types that are in close proximity and associated with response and resistance. We can then take this a step further and ask the question, what is the enrichment of two cell types being next to each other? And is there an association between response and resistance to therapy? And we find that the proximity of Tregs next to monocytes, or HLA-DR positive tumor cells next to macrophages, or Tregs next to CD8 cells is associated with response or resistance to therapy in this cohort. And so what we then did was, in collaboration with Nuclei from Israel, was we were able to benchmark our spatial multiplex IF data to pathology annotated H&E and spatial multiplex IF data. And so we have this sort of ground truth now that we can benchmark this pipeline of data to, and this is sort of the computational pipeline for that. And so we can do our multiplex imaging, we can do our cell seg, the cell typing, the neighborhood analysis, and so on. But remember, we can now subset based on the metabolic data. So this is just looking at marker performance across it, and so most of the markers have done well. This is just looking at the binary classification, and most of the markers have performed well here. And what I'll skip to is this is now you're using a lens of pathology annotation on the tissue, so a combination of supervised and unsupervised clustering. What you have at the bottom here is the unsupervised clustering, at the top is the supervised clustering, and you can see we can resolve the CD4s, the T cells and the Tregs, and the CD8s a lot better using the supervised approach. And so what's really important is we can now start to identify subsets within every cell type. So for tumor cells, for example, we have metabolically active tumor cells on the left and metabolically dim cells on the right. And then for the CD8s, by the same token, we can see these metabolically active tumor CD8s on the left, these metabolically dim cells in the middle, and then this third group, which is metabolically active. And when we go back to the imaging data, these are actually the CD8s that are infiltrating the tumor. And so the lens of metabolic activity allows us to glean more information of the microenvironment and the cell type subsets that are actually functionally responsible for clinical endpoints. And so we can look at this on a global heat map. The immune cells are on the left, the tumor cells are on the right, and you can see the markers that are helping us functionally annotate these: GLUT1, G6PD, citrase synthase, NRF2, and so on, PDL1, CD44, and so on. And so what we found was that there was this metabolic signature defined by increased glucose uptake and ASCT2 and NRF2 that was associated with a worse outcome to checkpoint inhibitor therapy, really linking into that Warburg effect. And so if we go on to a clinical trial, this is looking at a translational biomarker study, single agent immunotherapy versus standard of care, whole slide tissues. Again, remember I was saying, your single cell experiment is 50,000 cells. We're looking at 3 to 5 million cells now, spatially annotated. We can look at where the macrophages are, the pancytokeratin, the CD20s, and so on, the B cells, the MAX, the infiltrate, and so on. And then we can contrast between clinical endpoints of response to therapy. So we can contrast patients that have had complete response to immune checkpoint inhibitor and those that have had progressive disease. And what we find is that in patients that have a worse outcome to immunotherapy, they have increased glucose uptake, specifically G6PD and GLUT1, specifically in the tumor cells. Whereas in patients that have response to checkpoint inhibitor therapy, the expression of these glucose markers is a lot less. And so we looked at the RNA profile for these patients, and what was fascinating to see was that we had increased expression of markers involved in ferroptosis, specifically GPX1 and GPX4, in every contrast to response. So in every contrast of responders to non-responders, we could see increased elevation of markers involved in ferroptosis, linked to the non-responsive group to checkpoint inhibitor therapy. So it was clear in every clinical contrast. And so when we put this data together, we can now follow this. This is in a non-responder. This is where the tumor cells are in the red. Blue is low, red is high. So this is pancytokeratin. That's where the tumor cells are. CD8s, they're on the periphery. They're not infiltrating. Macrophages, again on the periphery, not infiltrating. GLUT1, so glucose uptake in the microenvironment, is all localizing to where the tumor cells are. G6PD, almost a gradient-like expression through the tumor, and fatty acid metabolism, CPT1A, localizing to where the T cells are. So you can now get this metabolic information overlaid with your cell type information. And this was really interesting because we saw this paper come out of AstraZeneca from Dmitry Gabrielovich's lab, showcasing the importance of ferroptosis in maturing MDSCs. And this is another study from a colleague of ours, DU, who's a couple doors down from me. And so if we move now to head and neck cancer, we've now applied this 580-plex panel of proteins that's been developed by NanoString and Abcam, which covers the hallmarks of cancer. And NanoString tells me that these panels are ever expanding, which is really exciting. And I'll talk to some of the really early work. We've actually submitted this work today. It'll come out in Research Square in the next 24 hours. And so this was in a head and neck cancer study looking at 85 patients, combining geomics, multicellular readout, 580 antibodies, and then validating some of the targets with single cell resolved proteomic approaches. This was sort of a discovery, a proof of concept study that'll come out in the next couple of days. And so we used a tissue array because we could screen about 85 patient samples, single core biopsies per patient. Then we could validate using low-plex proteomic aspects. And so what we did was we gated on tumor by cytokeratin and non-tumor by non-cytokeratin to effectively capture the tumor and the microenvironment. This is just looking at the library sizes to see that there's no confounding data here, going across multiple different groups here. And so what we can see, which is reassuring, was we could see that this is just all data QC up front. We could normalize the data using RLE. We could see that the tumor separated out from the microenvironment really clearly, the red and the blue. And the other thing that was separating out the classes at PCA was the groupings of the patient, so stage 1 to stage 4. And so we could then run these 580 antibodies against clinical endpoints such as disease-free survival and overall survival. And we could see that markers such as CD34, so high CD34 was associated with a worse outcome, and markers such as CD3 or high CD3 were associated with a better outcome. And we had this at the RNA level too. We then took this and we wanted to validate the CD34 findings independently. So we ran a CD34 antibody in combination with a number of other markers on the phenocyclofusion. And we could see that high CD34 indeed localized to where the tumor cells were associated with a worse survival. We then looked at the stromal compartment because remember, we've got the tumor and the stroma now, and we could see that high ADORA2A in the microenvironment was associated with a worse outcome, and high interferon gamma in the microenvironment was associated with a better outcome. So this is just taking a lens of discovery of 580 antibodies and being able to screen across multiple tissue samples and tissue arrays and being able to identify new targets associated with your clinical endpoints. And it was reassuring to see that more known markers and potentially unknown markers were coming up in this screen. And it provides us a framework and a workflow to be able to scale this to immunotherapy cohorts and so on. And so we now imputed the whole transcriptome data from this. And so we ran the IPA and the whole transcriptome, and we could look at the predictive signatures in both the RNA and the tumor compartment and the protein compartments. We then took it a step further and we looked at the correlations between the IPA 580 antibodies and the whole transcriptome 18,000 genes. And what was really reassuring to us was we could see high concordance, almost 0.9, with the keratins and a number of structural markers, whereas the correlations were a lot less comparing between RNA and protein, sort of involved in functional or immune cell types. So this was really reassuring that we could see that even at the contrast between RNA and protein. And so I'll go into head and neck cancer before I go into some of the COVID work. This is in head and neck cancer. This is in a patient that had a partial response to immunotherapy, died after 14 months. We had the pre-surgical resection, and we wanted to see the microenvironment and see whether we could identify the heterogeneity within the microenvironment. So we stained the serial section with 101 antibodies, serial to what the pathologist collected, based on the hallmarks of cancer. And what we found was fascinating. We could see the tonsillar regions, we could see the tertiary lymphoid structures within the microenvironment, and this one tumor could be broken down into multiple subtumors. So tumor regions 2, 4, 1, and 3. And so you could see these normal germinal centers in the middle. This is in head and neck cancer, HPV positive disease, and you had these tertiary lymphoid structures infiltrating the tumor at the top, whereas it was absent in tumor regions 2, 1, and 3. We could then localize all these cell types, the B cells, the CD4s, the CD8s, the DCs, the M1s, the M2 macrophages, and so on. And we could see that these tertiary lymphoid structures were mature. They had high CD20, 21, FOXP3, and PD1. We then contrasted the TLS at the top that was infiltrating the tumor to these normal germinal centers that were distant or non-tumor associated. And you can see the differential expression at the whole transcriptome level between the TLS and the germinal center. They're clearly different, and they're also driven by this interferon pathway. We can see this at the whole transcriptome level by contrasting on pathology-informed structural features in the tissue. This isn't spot-based data, it's pathology-informed tissue data, and FFPE too. And so we could then identify that the M1 macrophages were all localizing to the bit in the yellow where the TLSs were. The M2 macrophages were localized to the bit in the purple where there was an absence in these TLSs. And so on 2 dimensions, we could see this partial response to checkpoint therapy, the presence of TLSs in one region and the absence of them in the other regions, but also this M1, M2 polarity. What was really telling for us was when you looked at the glucose uptake and a number of metabolic markers, we could see that there was increased glucose uptake in tumor region 3. It had increased expression for G6PD, whereas in tumor regions 4, 2, and 1, you had increased expression for pro-apoptotic markers or markers involved with cell death. And so what you're seeing in the same tumor is areas of sensitivity at the top defined by pro-apoptotic markers, tertiary lymphoid structure infiltration, maturity of TLSs, and M1 macrophage biology, whereas in tumor region 3, you've got high glucose uptake, an absence of tertiary lymphoid structures, and an M2-like macrophage phenotype. So what you're potentially seeing is sensitivity to checkpoint at the top and resistance to checkpoint at the bottom. And if you build the spatial metabolic map, what you have is this area of resistance defined as tumor region 3. On the periphery, you've got fatty acid metabolism and DNA damage, surrounded by areas with increased expression of MMP9, which is involved in EMT. And so this is the likely invasive edge of the tumor, whereas all of tumor regions 1, 2, and 4 are dead, dying, or necrotic regions. And so you've got pro-tumor and anti-tumor activity in the same tissue. And so I'll talk about this really exciting, this is brand new data. This is looking at, so everyone infers proximity, everyone infers receptor ligand interactions at the moment. We can now visualize that using proximity ligation work. And this is comparing in head and neck cancer between 32 patients that have received immunotherapy, between responders on the left and non-responders on the right. This is just contrasting two. We've got this for the entire cohort. And we can cycle through actual interactions for PD-1, PD-L1 by proximity ligation, where you have rolling circle amplification that can be fluorescent, and you can visualize that. And so we can, when we contrast, we have the complete responder at the top, the partial responder in the middle, the progressive disease at the bottom, and the stable disease at the bottom right. What you can clearly see is you've got on the left cell typing neighborhood, and then PLA signal or proximity ligation signal in the purple is the difference in the staining pattern of the interactions between PD-1 and PD-L1, between the responder and the non-responder. Here we've got this for the whole cohort. The study will be submitted in the next week. It's really fascinating to see where the actual interactions are happening, in contrast to PD-1 or PD-L1 protein expression. And so we've got activity in skin cancer. Skin cancer is a really good organ because you can map these across multiple time points. We're collecting tissue at progression and at cure. And so we can contrast between patients that are sensitive to immunotherapy and skin cancer versus those that are recurrent. And we can start to look at things such as the differences in composition, but also metabolic activity, which I won't spend a lot of time. But what was fascinating is now in skin cancer, we have multiple biopsies over the course of therapy. So we can now measure what are the changes in the microenvironment, factoring in time between responders and non-responders. These are in the same patients where we're getting a punch biopsy over the course of immunotherapy. And we can start to find proximity-based differences between the baseline and the follow-up sample in skin cancer, all factoring in the time component into this. And so I'll finish off with the work in COVID, which is super exciting, where we've run a number of studies, well published in lung, cardiac. The brain study will be submitted soon, and the placenta studies published, where we were looking at where the virus was infecting cells in the lungs, and we could identify this by RNA-scope. We then ran multiple NanoString assays over this, including the whole transcriptome and the CosMx, the 1000-plex RNA panel, to really resolve this signal. And we could contrast between COVID and flu and normal healthy lung tissue or non-viral deaths. And so this is the computational pipeline for data QC that Ning's published earlier this year. So this is a really good resource to do sort of genomics-based analysis if you're looking for a tool for that. And so we could then contrast between COVID-19 lungs and control in COVID-19 lungs and H1N1, the 2009 pandemic. And what we found using the geomics was that IFI27 and Ly6E were two top differentially expressed genes between COVID and control and COVID and flu. This is a type 1 interferon response that we're detecting in the lungs of patients collected at rapid autopsy. When we looked at this study, we could see that IFI27 and Ly6E were conserved across multiple lung autopsy studies, and it was really interesting, but we couldn't localize this to a single cell resolution at the time. The study was done in 2021 when we didn't have the tools, but then NanoString came out with the CosMx spatial molecular imager, which allows us to go in and look at single cell resolution information and subcellular data, right? And so we're really excited by this new platform. It allows us to localize transcripts per cell and subcellular readout. And we looked at an independent cohort of patients. So we had TMA A, B, and these normal control lungs. So we had about 40 patient core biopsies taken from the lungs, and we ran this 1000-plex RNA CosMx assay on this. This paper was published by a colleague of ours, and we're all on this study. And so we could localize where the transcript, where the virus was by RNA-scope. We could then overlay that with the multiplex IF data and then the spatial molecular information on the right. So we could localize the virus and the single cell data at the same time. So what does this look like at a high resolution? So we can see here in this area of viral infection, you can see this red region on the top left. These are transcripts that we can now map. Every dot here represents an individual transcript for CXCL10 in the yellow, IFI27 in the red, and so on. And so what we found was that the macrophages were localizing to the sites of viral infection, and that's where IFI27 was coming up. So we could contrast viral regions to viral negative regions and localize that this was coming from the macrophages. And so we submitted this work to eBiomedicine, and the reviewers came back and told us that there are 10 different types of macrophages, which ones were they? And so the beauty of having 1,000-plex data is you can go back and mine that. And so we could identify the alveolar macrophages, we could find the interstitial macrophages, we could identify the recruited macrophages, and localize where IFI27 was coming from, and it was actually in these alveolar and interstitial macrophages. And we could actually report on this, and then they accepted the paper. So this is the benefit of having the higher plex assays, which allows you to deconvolve a lot better. And hopefully with the 6K and the whole transcriptome panels coming out, those are going to help us get more granular into this information. And so then Tony in the lab, who got really frustrated with cell types, he said that what if we were to just look at cells in space? And transcripts in space. So leave the cell typing out. Let's just look at mRNA transcripts in space. And can we look at the distances between transcripts and the location of the transcripts? So he looked at 100 million locations in the same cohort, 100 billion distances in the same data set, and he computed the information. And what we could see was that the transcripts were, in fact, traveling to, so the transcript species were found to be in similar areas of interest. So the heat shocks, for example, were found in similar regions of interest, similar areas. The interferons, the HLAs, and the HSP90s, all these families of transcripts were found closely associated. He found 107 pathways or super pathways that passed FDR. And so when we looked at the pathways, they were involved in interferon gamma, they were involved in IL-6, they were involved in fibrosis. So these are known pathways that we could identify using cell-based approaches, right? So what's the novelty of this? We could also identify that these were SARS-CoV-2 specific, which is nice. What was fascinating was we could start to see, using a cell-free approach, that oxygen CO2 in red blood cells were being perturbed during COVID-19. You would not be able to do that using a cell-based approach. The other pathway that was perturbed, which was interesting, was this pathway associated or perturbed in the nerve cells found in the lungs. And so you would never be able to identify these or be able to cell segment these based on cell-based approaches. So we think this is a really neat approach to getting more information from using a cell-free analysis of CosMx data. And we're starting to overlay this with a lot of public analysis and experimental data. And so what I'll finish with is, we had this really crazy experiment, and you heard Joe this morning. We had a colleague, Kirsty Short from UQ, who came and said, look, we've got these collaborators in Germany and Austria who've got archival tissues. These are Spanish flu tissues collected over 105 years ago. Can we do spatial on them? And so I thought, what would Joe Beecham say? And I said, let's do it. So we had a student spend a year in Germany and Austria collecting these tissues. Again, we reached out to about 60 labs. 59 said, no, you're not going to get anything from them. One said yes. So we got 8 lungs collected. And we ran the 570-plex protein and whole transcriptome over these. This is the PCA of the protein data showing separation of your exudate regions of interest from your pneumonia regions of interest at the protein level. This is the differential expression between the exudate versus pneumonia regions. So the protein data is beautiful. It stands, it holds. This is the RNA data. So this is on PC1 and PC2. You can see the transcript data separates out based on the histology. So what you're seeing is real histology separating out your transcriptomic data. Your exudates in the red, your hemorrhages in the green, your pneumonia regions are in the blue, and your transudates in the purple. So even at the RNA level, The histology separates out the transcript data. This is the RNA differential expression between two of those groups. It's a bit wobbly, but we've got a couple samples that are really good. And so we then ran the protein assay over this, and we could, from about a 21 plex panel, we could see good signal across about 8 markers, including macrophages, B cells, and so on, which is really exciting. So RNA and protein data of tissues that are over 105 years old. It made the cover of the Australian, and it talked about cool science. This is Lauren, who spent a year in Germany collecting these tissues. I'll encourage you to come down to Multiomics in December 1st to the 3rd. We have some incredible speakers across the US, Europe, and APAC regions coming down for this meeting. It'll be a stellar meeting, December 1 to the 3rd in Brisbane, Australia. Welcome all of you to come. It's a beautiful city, and Joe and a lot of the others will be down for the meeting. And I would like to end by thanking the patients that have given their samples to these studies, the collaborators who've put timeless effort, the lab that does a lot of the work, the computational folk dotted around the world, and the funding agencies that support our research. And I welcome any questions. Thank you.